Thank you very much for giving me the opportunity to talk today. I'm very happy to be here. So I'm going to be talking about genetics, and some of what I share has already been talked by other speakers. Probably is going to be a refresher. So a little bit about gastric cancer, not that it's not been touched already. It's a third leading cause of cancer death worldwide. There's 700,000 deaths occur every year. It's a heterogeneous disease. I mean, it means it's not one disease, it's probably more than one disease. We tend to study this just as one disease, which needs to change. It's an aggressive disease. So a little bit about what we know so far. 90% of all gastric cancers are sporadic gastric cancer, which means we don't, there's no particular inherited mutation that is responsible for that. 10% is familial, and 2 to 3% is hereditary. Gastric cancer has been suspected to have some familial basis, for a long period of time. This is Napoleon Bonaparte, which you might all know about his story in gastric cancer. This is his family tree, and there's here Napoleon, who died at 52 with known gastric cancer. His father had gastric cancer. His grandfather was suspected to have gastric cancer too. So was his one sister, and 2 other sisters, and 1 brother was suspected to have gastric cancer. That's pretty impressive. Though some French don't think that he died of gastric cancer. They think the British poisoned him when he was imprisoned in South Atlantic. Anyway, familial gastric cancer means the cancers that are observed in the family, but they might be in family, but they not be truly hereditary. Truly hereditary cancers would be either the patients who have CDH1 mutation. We're going to talk about that. CTNNA1 mutation present similarly to CDH1 mutations. They give rise to what we know as hereditary diffuse gastric cancer. It's a very aggressive gastric cancer for which we have very few treatment options. There's also recognition of gastric adenocarcinoma and proximal polyposis of stomach syndrome. The mutations for this is not identified yet. Also not identified is a mutation in familial intestinal gastric cancer. We do see increased incidence of gastric cancers in other syndromes, most prominently Lynch syndrome. We also see increased incidence of gastric cancer in familial adenomatous polyposis syndrome, Leigh-Fermani syndrome, and Peutz-Jeghers syndrome. These 4 syndromes give rise to other cancers, not just gastric cancer, whereas these hereditary gastric cancer just give rise to gastric cancers. Though in CDH1 mutation, you also see lobular breast cancers in women with CDH1 mutations. Another example of how gastric cancer can be seen in family, this is the original family history of CDH1 mutation. This was originally diagnosed in a group of New Zealand Maori people in New Zealand. You can see gastric cancer presenting in many patients in the family. So let's talk more about that one. CDH1 mutation leads to loss of expression of cell adhesion molecule E-cadherin. It keeps the cells together. When that is lost, they tend to migrate very easily. It's transmitted in an autosomal dominant manner, and it has high penetrance. When I say high penetrance, means if you have a mutation in the family, a lot of people will get the cancer. In fact, by age 80, 70% of men with this mutation will have gastric cancer, and 56% of women will have gastric cancer. And they tend to present early. Average age is about 38, which is about 20 years younger than otherwise. As I said, if a woman with this mutation can also develop lobular breast cancer. If you have a family, if you have one person diagnosed with this particular mutation, you need to screen the entire family. And if anybody has it, they should have prophylactic gastrectomy by age 20. Okay? One thing about gastric cancer, as I said, this is a heterogeneous disease. There's probably more than one cancer, is what we're calling, all by a single name called gastric cancers. And it's very important to, in the future, recognize the genetic complexity and identify subtypes of gastric cancers and treat them accordingly. Traditionally, we've classified gastric cancers as either intestinal or diffuse. Intestinal means you develop glands like intestines, whereas diffuse, you develop these kind of cells, the signet rings, and there's no proper gland formation. And this particular classification has been used for many years, 50, 60 years, but this really has not been able to guide us to sufficiently improve the outcome of our patients. So there is a need for better molecular classification of gastric cancers. And Dr. Ajani has already mentioned about this, and so has some others. The first comprehensive data about molecular classification came from TCGA. There was 298 patient specimens that were taken, and with that, there was very comprehensive genetic analysis done, including whole exome genome sequencing, microRNA sequencing, and messenger RNA sequencing, and all others. And based upon the kind of genomic profile they have, 4 different kinds of gastric cancer was proposed. This is molecular classification of gastric cancer. In each subtype, there was a predominance of different mutation. For example, in EBV positive gastric cancer, about 80% of the patient had PIK3CA mutations. There was also PD-L1 overexpression. PD-L1, as mentioned by others today, is the one that will indicate possible response to immunotherapy. This particular cancer might respond very well to the immunotherapy that was just mentioned. The next class is the microsatellite unstable class. This is the one which has MLH silencing, and we have already talked very extensively on this particular class of gastric cancers. And they are also the prime candidates for immunotherapy. The role of immunotherapy in these 2 classes is probably not going to be substantial. There might be some in CIN, probably not much in genomically stable group. The the hereditary diffuse gastric cancers belong to this category. That's a genomically stable category. There is CDH1 mutation and also RHOE mutations occur in this particular subgroup. While this particular classification was able to molecularly classify the gastric cancer in 4 different types, there was not much of a survival difference in all the 4 groups. So while this particular data was interesting, it was not helping us to see if there's a survival advantage between those 4 groups. And another important study that came later was from actually the same year in 2014, again published in Nature Medicine, was data from Asian Cancer Research Group. 300 patient specimens were taken, and there was genomic expression analysis done, and based upon that, there was 4 groups of gastric cancer classified. And the interesting thing about this particular criteria to classify gastric cancer was that there was not only a molecular basis for classifying these 4 the cancers in 4 different groups, there was a survival difference between each group. I'm now going to talk about that in the next few slides. So this group also had the microsatellite unstable. This group tend to have the best prognosis. They tend to occur in early stages, and not many of them would have metastatic disease. The second group is the epithelial mesenchymal translocation group, EMT group, and this was the worst prognosis. This is the one that has more, this group has the CDH1 mutations. They have this diffuse histology. 80% had diffuse histology, but only 24% of all diffuse cancers were of EMT subtype. And they frequently had peritoneal metastasis, and distant metastasis was rare. So important thing to notice is not only they have a worse prognosis, they also tend to have just peritoneal metastasis. So this probably is the worst group that we frequently clinically observe. We struggle with. We wonder what's happening with these patients. And this particular subgroup needs to be studied more in a different way than what we've done so far. Microsatellite unstable group, we already discussed about, microsatellite stable and P53 intact group tended to have the EBV mutations, EBV infections, and they also tend to do very well. And this group, the microsatellite stable and P53 loss, probably represents the bulk of gastric cancers, and they probably are the ones who respond favorably to the chemotherapy. This is the survival difference that I mentioned about. As you can see, each of these 4 groups tended to behave differently. And this is from the original Asian Cancer Research Group data. And this data was replicated in 3 other cohorts, as well as the TCGA cohort. And in every cohort, there seems to be a survival advantage when we use this criteria to classifying the disease. So in conclusion, gastric cancer is a heterogeneous disease. The one-size-fits-all approaches are likely to result in better outcomes. The future clinical trials must recognize this heterogeneity and should be geared for individual subtypes. And what we need is, we need this subclassification to be available commercially so that they can be implemented in both clinical practice and clinical trials. And we have to recognize there is a hereditary gastric cancer, even though they contribute only to 2 to 3%, they tend to have bad prognosis, and we need to consider prophylactic gastrectomy by age 20. Thank you very much.